In Chapter 13, we go through the different types of mutations that we might find affecting genes, and also how mutations are caused in cells and how they are repaired in cells. Unfortunately, from a lecture standpoint, the first video is usually just kind of a laundry list of types of mutations, and many of these are mutations that we have seen already from different illnesses or diseases that we have seen throughout the semester. Others are going to be kind of new. And then after we get through the types of mutations, then we'll do a short section on what causes mutations and how cells repair mutations. Our example here of Lou Gehrig and Stephen Hawking, these are individuals that have Lou Gehrig's disease, or ALS, which is amyotrophic lateral sclerosis. It is essentially a neurodegenerative disease that destroys motor neurons in the brain. And so because these motor neurons are being progressively destroyed, it leads to muscle weakness, paralysis, and respiratory failure. The reason that we're talking about this here is that it is caused by expanding nucleotide repeats. This is basically repeating nucleotides, a small number of nucleotides that get progressively longer and longer and longer. So we're going to talk about that type of mutation and how that might come up, along with some other types of mutations that we commonly find in cells. At this point in a genetics semester, you're probably well aware that a mutation is a change to the genetic sequence of an individual. And in a genetics class, we are particularly interested in inherited genetic changes. Very often we may encounter diseases that are not inherited, such as skin cancer, or other types of cancer that are in fact mutations to the genetic code, but are not necessarily passed down from individual to individual because those alterations occur during their lifetime rather than being encoded in the genome that gets passed on to the next generation of cells during meiosis. So generally, mutations tend to have a negative effect on an organism. And this is because an organism is already adapted to the environment that they're in. Their set of genetic information is probably specific to the environment, to the requirements of that environment. And so an individual is probably already well adapted to whatever environment they find themselves in. However, mutations are not all bad. Sometimes mutations are actually good and they provide an advantage to an individual. Other times mutations are neutral, so a change happens, but we don't really see a phenotypic effect. But one of the key things about mutation is that it is the ultimate source of genetic variation. So we have talked in the semester about how genetic variation comes from the process of meiosis and recombination and independent assortment, combining of genomes, all of that. But mutation is the only way that we get new variation. This is as opposed to shuffling the current variation. So with mutation, we can end up with brand new alleles. Now, sometimes they're good, sometimes they are not good, sometimes they stay in a population, other times they don't. It just depends on how evolution works on those mutations. There are many types of diseases and disorders that result from mutations in humans. We've had several examples throughout the semester when we've talked about different types of phenotypes and whatnot. Mutations also useful for understanding biological processes. So one of the things that we can do with a mutation is to use it to figure out what a gene does. So we can do something called knockouts. And I apologize, my pen is not working for some reason at the moment. We can do something called a knockout, which is where you disable a gene and use it to figure out what effect the dysfunctional gene has, because you can then work backwards to figure out what the function is supposed to be. So you can use a mutation in an individual as basically a diagnostic tool to figure out what part of any biological process is being controlled genetically. There are two broad categories of mutations. Those mutations that occur in somatic cells, or our body cells, our non-sex cells. And these mutations are important for the individual, but from a genetic standpoint, they're not as important. And I only say that meaning that they're not important from an inheritance standpoint. Absolutely, a somatic mutation can cause something important in the individual. But as far as passing that mutation on, somatic mutations will not be passed on to the next generation. However, when a mutation occurs in a germline cell, such as our sperm or eggs, these ones are important from an inheritance standpoint because they are going to be passed on to the next generation. So if we have a mutation in our gonad cells, and remember our gonad cells produce the sperm or the eggs, if we have a mutation that happens in the gonad cells, then that mutation is going to end up in any sperm or egg that is produced from that line. And so these ones are going to be passed on and part of a cell line. So they are going to be passed on through a family line. They are going to become part of every cell in an individual that is made from one of those sperm or eggs. Earlier in the semester, we talked about chromosomal mutations. In this chapter, we're mostly going to focus on gene mutations. A gene mutation is a sequence mutation, so a change in the sequence of a gene. And this is in contrast to a structural mutation. We talked about structural mutations in chapter 6. We talked about things like deletion of a piece of chromosome or a section of a chromosome. Likewise, we talked about duplication, inversion, translocation, and also a couple other things like fragile sites and expanding repeats, things like that. We are going to touch just very briefly on expanding repeats again, but usually repeats in a genome are going to be on a smaller scale. So we are going to focus on these gene mutations, or changes in sequences. So if we have a gene mutation that happens in a somatic cell, then this mutation, found here on the skin of the fish, is only going to be found in any cells that come from that specific region of the body. And so it's only going to be found in this particular fish. In this case, it's on the skins or the scales of the fish. And so since it is not being found in the gonads, then it's not going to be passed on. If, however, we get a mutation in a germline cell, say our sperm or our egg, then any sperm or egg cell that is produced from that original cell is going to carry that mutation. That means that every single cell in the offspring of that sperm or egg is going to carry that mutation. In contrast, an individual who acquires a somatic mutation will not pass it on to the next generation of individuals. So their gametes are not going to be affected by this mutation. So once we have a mutation in a germline cell, we can start to look at what type of mutation actually took place. There are a couple of broad categories here. The first is a base substitution. Now this is where a single base or a very small number of bases is substituted for a different base pair. Now generally this is going to be due to DNA replication. So a mutation occurs or an error occurs during DNA replication. our DNA proofreading enzymes, DNA polymerase 1, or whichever the case may be, doesn't recognize that change, and it sort of slips through and gets passed on to the next generation. So there are two types, transitions or transversions, and we'll define that on the next slide. We can also have insertions or deletions. And again, these are gene mutations at the base pair level. So this is going to be a small number of inserted or deleted nucleotides. And here we can have two different options. One is a frame shift mutation, where this is going to cause a change in our reading frame. We talked about reading frame in chapter 11. This is the triplet set that we read based on our messenger RNA. Remember, these are non-skipping, non-overlapping triplets. So we set our reading frame based on the very first AUG. If we insert or delete something, that is going to shift our reading, shift our triplets, and change our amino acids. This happens when we insert or delete one or two nucleotides. If we insert 3 or a multiple of 3 or delete 3 or a multiple of 3, this is called an in-frame insertion. And what this means is that we insert one codon. So a whole triplet, we insert a codon, but it doesn't cause a frame shift because we've just inserted 3. Yes, it does insert an additional amino acid, but everything around it or after it remains the same sequence. And then we've got expanding nucleotide repeats. This is an increase in the number of copies of a small set of nucleotides. So this is just a few nucleotides. So it might be a repeat GC, 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 etc. And maybe during the next round of replication, we get a little bit of trouble. And instead of having 5 repeats, maybe we increase it to 7 repeats. And then maybe the next time we increase it a little bit more. So that's what we mean by expanding nucleotide repeats. Again, usually a small number of nucleotides here. There are two types of base substitution mutations. The first is a transition. Now, in a transition, we are going to substitute a purine nucleotide for a purine, or a pyrimidine for a pyrimidine. So in our example down here on the bottom, we have an A transitioning to a G or vice versa. Remember, the A and G are both purines. C, T are both pyrimidines. So on the bottom, we have a T transitioning to a C and vice versa. In a transversion mutation, we have a purine and a purine being substituted for a pyrimidine or vice versa. Now, when we have this substitution, we are actually going to affect the structure. Because remember that a purine is 2 rings wide and a pyrimidine is only 1 ring wide. And when they pair up, then they are pairing up in a 3-ring across structure. And this is one of the ways that our DNA molecule maintains a standard width all the way across. But if you are supposed to have a purine and you substitute a pyrimidine, now you have only 2 rings across. And so you can actually affect the DNA physical structure by having a little skinny spot. Or if you insert a purine, you're going to widen the structure. And this is going to cause some problems for our replication and our transcription processes. If you do any sort of base substitution, there's always the potential of causing a missense or a nonsense mutation. And so, not that there wouldn't be a problem if we have a transition mutation, because either type, transition or transversion, can potentially change that amino acid. We just don't get the structural change in the DNA with a transition mutation. So we've already briefly explained what an expanding nucleotide repeat is. This slide explains how it increases the number of nucleotide repeats. So if we look at the top left, we've got GTC, 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 etc., and then the complement sequence under that. And remember that during DNA replication, our strands are going to separate, and then we're going to replicate one side. However, because these are repeating nucleotides, and because of complementary base pairing, if any strand happens to pull apart and slip, it can pair within itself, and we end up with this hairpin turn like we saw in the structure of RNA. And so if this happens, if that little hairpin turn comes out and the DNA molecule binds to itself, then what happens is that even though we've already replicated, this purple strand is our replicated strand, but when it comes off and binds with itself, it leaves a region that's already been replicated, but the cell doesn't realize that because it's exposed and opened up the double helix. And so this region here gets replicated again. And what we end up with then is a longer sequence the next time it goes through replication. So instead of having 8 nucleotide repeats, once it backs up and we accidentally replicate it twice, then we have 5 extra. And so we've expanded from 8 repeats all the way out to 13 repeats. And so anytime the strand slips and binds to itself, then it's going to accidentally double replicate these pieces. So we have talked about the cell types that can acquire mutations. We've talked about the sequence changes that occur in a mutation. Now we're going to talk about the phenotypic types of mutations, or the phenotype effects that we might have from a mutation. So again, this is just kind of a laundry list of different things we could have. A forward mutation is where we go from a wild type to a mutant type. Now this could be a new mutant, or it could be an old or a known mutant. We shouldn't really say old. Or it could be a known mutant. So you can go from normal to something new, or normal to a different mutation that you've already acquired in a population. A reverse mutation is the opposite. This is where the wild type mutation comes back. So we have a mutation and the wild type is restored by a second mutation. Loss of function mutation means that the protein stops functioning, either reduced function or completely lost its function. Gain of function mutation can happen in one of two ways. Either the protein can acquire a new function, or its current function becomes increased. You produce more of the protein, and then you get basically a higher output of the function of that particular protein. In a conditional mutation, the mutation shows only in certain conditions. So if you don't have those specific conditions, you see the wild type. Under those specific conditions, you see the mutant type. A lethal mutation is a mutation that causes death in the individual. This is usually early on, so the individual doesn't even really survive. Of course, it could be something that happens a little bit later, but usually we talk about lethal mutations as something that happens early on, so that you don't even see the number of individuals that you would expect with a particular phenotype. Often this is going to be a sort of a lethal in one situation. So sometimes it is lethal no matter what. So it's lethal if you are if you have the mutation. Sometimes it is only lethal if you have 2 copies. So maybe it is a homozygous dominant that is lethal, but the heterozygous is not. So there's a couple different ways you could see the lethal mutation. On the next slide, we have something called a suppressor mutation. And this is a situation of epistasis. We talked about epistasis way back at the beginning of the semester. This is where one gene controls the expression of another. And so in a suppressor mutation that is showing epistasis, this is where a mutation at one gene is covered up by a mutation at a second gene. And so instead of seeing the first mutation, you see the wild type because of the second mutation. I'll show you an example. We have examples of forward and reverse mutations. So we are all familiar with the red eye wild type in fruit flies, this guy here. So he's got the normal or wild type mutation for red eyes. A forward mutation in, say, gene A is going to produce the white eye, which we are also familiar with at this point. A reverse mutation takes the white eye back to normal red eyes. In the second step here, we see a suppressor mutation. So we have, in this case, the mutant white-eyed allele, but we see it as normal because this second mutation, the B mutation, actually restores or takes away the mutant phenotype at the first allele. Sorry. So we have one allele controlling the expression of the other mutant phenotype. So lastly, we have the effect of our mutation on the amino acid sequence that we are producing. And of course, this is going to affect the protein function. So if we have a change in that amino acid, it's going to affect the protein shape or structure, which in turn affects the protein function. So there's a few different ways that we can see changes in our amino acid structure. Remember, this is based on our nucleotide structure and then our codon reading and then the linking of our our amino acids into a polypeptide. A missense mutation is where we insert a different amino acid. So we have a mutation that causes a messenger RNA that codes for a different amino acid at one place. A nonsense mutation is the introduction of a stop codon. So we truncate our protein. A silent mutation is one that causes no codon change. So this is due to degeneracy. In our codons. Remember that there are multiple codons for most of our amino acids. So if you just change from one codon to another that still codes for the same amino acid, this is going to have no effect on the protein whatsoever. Last is a neutral mutation. A neutral mutation has an amino acid substitution. So it is in fact a type of missense, but there's no change in function. So this is usually because something happens like a polar amino acid is substituted for a different polar amino acid. And so because you keep that polar molecule in there, you're not really changing the chemical behavior of the protein. Pardon me. And so this doesn't really affect the function, which means, the structure, which means it doesn't really affect the function.